So let's start a new conversation, okay? Exams are graded and posted, and I gave them to my secretary, and I haven't seen her today, so I don't know what's going on. But did anybody try to go by and see their exam? Not yet. They are there for you. Exam, considering all the lovely looks you were giving me on the way out and during the exam, it went actually quite well. And I didn't curve it as much as I thought I would have to. Right? So that's good. Right? Only 5%. Only curved at 5%. So that's good. Right? That's a good sign. And there were only, I think, 4 people who were below water. So in a big group with a lot of people learning, I think it was a successful exam as far as your performance in it. So I want to congratulate you. There was a lot of material thrown at you. The hormones are always a tough road. And in the end, you may not feel like you got it, but you got it well enough to move forward. Our next exam will be after spring break, and I know that's all any of you are thinking about. There's no 106 exam this week. Notice, no one's here. There's no 106 lab exam this week. Notice. How many of you have lab tonight? Yeah, a lot of you, right? That's why you're here. And maybe you wouldn't have been here otherwise. I don't know. But the next exam will be after break. Right now it's scheduled for St. Patty's Day. I think it's on Tuesday, March 17th, if I remember right. That gives me 4 lectures to talk to you. I may, you know how I hate moving exams, but because I moved the last one, now this one might get moved. I've got to figure this out. I've got, on the next topic, we're going to talk about muscle. We're going to be talking about muscle physiology and actin and myosin and how that whole thing goes together and works. Then we'll also talk a little bit about cardiac muscle, and that'll bring us into the cardiovascular. So the next 2 chapters, one on muscle, mostly skeletal, but a little bit of smooth and cardiac muscle, and moving into cardiovascular. The question is whether or not I'll have time to talk about blood or not, and whether or not blood will be on the exam or not. So that's kind of what I'm kind of tossing that right now. Thank you. I'm kind of tossing it up and down right now, and I'll give you word very soon by the end of the week so that we can plan ahead. So the exam might get moved to the 19th, Thursday the 19th, to accommodate the blood. The blood really does fit better with cardiovascular as a topic with that content. This is a long chapter, and a lot of it you're familiar with. Some of it you're not as familiar with, and some of it's brand new. And so I want to just briefly think about skeletal muscle and get us thinking about this. You already know the types of muscle, right? You already know cardiac and smooth and skeletal. You know which ones are voluntary and involuntary. You know what they look like under the microscope. Some of this will be very quickly reviewed just to get your mind back into the muscle world. And then we'll move into some things that probably are not familiar to you. The 3 chapters for this exam are 9, 19, and 17. Sounds right to me. 9 is muscle. 19 is the cardiovascular. I will, by the end of the week, post a quiz on mastering over skeletal muscle. Last time I kind of shorted you. I only had 1 quiz for you on mastering. I know you're really disappointed. This time I'll definitely have at least 2. There'll be 1 over muscle, and there'll be at least 1 quiz on mastering on cardiovascular to help you get ready for the exam. So there'll be 1 that I'll get done this week, and I'll post it along with some homework, and then you'd have over spring break if you're around to kind of look at that quiz and kind of let that all sink in nicely. When we get back, we'll be talking about the heart and cardiovascular. At that time, I'll post another quiz over that content. So there'll be at least 2 quizzes for this next exam. Any questions just about logistics or anything about syllabus or schedule, anything else? Yes, ma'am.
There was the email that said something about case studies.
Thank you. I did post the case studies. The case studies are a group activity, ideally done by 3 to 4 students. Most of you in your labs are working or have been working on your lab project in groups of 3 to 4 students. It is a natural place to turn to do the projects. I'm not saying anything, but some groups are working really well together and some groups are not working well together. And you're probably thinking, Mac, don't make me work with that person anymore. They're driving me nuts. I'll let you guys as adult learners work this out. If you want to use your lab group, I think it's more convenient. You already see them once a week in lab and you can chat a little bit. If you don't, that's okay. But make sure you're not leaving anybody all by themselves, okay? Just be good and kind to each other. The case studies are pretty straightforward. When you go onto Blackboard, under lecture materials, you'll see a case studies folder. Underneath that, for the case studies, the first set of case studies, there's 2 within that you will do as a group. And there's 2 to choose from. There's a group A or group 1, and they're all about scientific method and skeletal muscular. And then the second group, I believe, is about endocrine and cardiovascular. And you will choose one of the case studies from each group. Read through it. They're 2 or 3 pages long. They're not terribly long. And then there'll be some questions asked along the way. All you'll do is type up the questions. Most groups simply distribute and say, you know what, you do those 3 questions, I'll do these 3 questions, you do those 3 questions. And then before the deadline, come together, pull it all together, review each other's work quickly just to make sure no one made any silly mistakes, and then upload that to SafeAssign and give me a hard copy. So it's not a huge amount of work, to be honest. They're worth 25 points each, so there's a 25-point one now and 25 points later in the semester. You might want to take a look at that, and maybe even, if you're working with a group, assign sort of what's going to happen, or maybe at least look at those case studies and decide which ones you're interested in doing. Other people will, if there's a group of 4, 2 people say, you know what, I really like that topic, and 2 people say, I really like that topic. And then you each split up those 2 case studies, right? So there's 2 case studies to do in each group, and there's 3 to 4 of you. You can decide how to divide up the work amongst yourselves. Did I, in that email, suggest a deadline? Or I just said we talk about it? We talk about it, so let's talk about it. It was like now, right, or something? Let's push it out. Let's have the case studies due. They're really not that much work. I really don't want to have it the same week as the exam. I don't want to push it out too far. What is the Tuesday? Tuesday is the 10th. What about the 12th of March? That gives you this week, it gives you spring break, and it gives you the week when we get back, okay? So within the next 3 weeks, you should be able to find each other and answer a few questions with each other. If you have any questions, let me know. You could bring that to lecture on the 12th. You could bring that to lab upstairs on the, did I say the 12th? Yeah, the 12th, right? Is that Thursday? Thursday the 12th? So let's make a lecture time the deadline for that. Or let's say lab time. Lab time, 6:15 on the 12th will be the deadline for the case study number 1. So make that change in your syllabus. Thank you for that reminder. Was there any other business for me to deal with? Yes, sir.
What's the curve based off of?
The curve is based off the people scoring on the high end as well as overall class average.
Okay, so somebody got like a 90 bump up to 100 then?
That would be a 10% curve.
Okay.
So nobody got 100.
Okay.
Okay. Close? I don't think anybody got 100. Right? Close. Okay. Yeah, I just kind of gave a little bit of a bump based upon the overall class average, and I don't believe somebody has to make 100. Sometimes they do. Sometimes they make 104, 105. Crazy people. We just don't care about those people, right? They're freaks. We don't worry about them. Okay. No. Okay. So we don't care about the curve. We don't care about what they made. I care about what I made, right? That's what matters. Okay. Let's take a look at muscle. I'm telling you to look over the first couple units. The first couple units are, oh boy, there's 3 kinds of muscle. And you know what they look like. Go back and look over their modality. Are they voluntary or involuntary? And you know what muscle tissue does for you. I'm just going to blow through this. I gave you the slides so you could read through them. Remember that muscle, skeletal muscle, has 3 layers of connective tissue associated with it. Remember this story? There's endomysium, perimysium, and epimysium. So review that and review tendon structures and how that connects with bones. And remember that a muscle cell is a very different kind of cell. And so there's some unique vocabulary provided for describing a muscle cell. Things like sarcoplasm. Sarcoplasm is simple. Sarco means what? Muscle, right? So sarcoplasm is simply the cytoplasm of a muscle cell. The sarcolemma, we know that lemma means rind or peel. So around the outside of a muscle cell, there's a cell membrane. It's simply called the sarcolemma. Lots of mitochondria. Remember, we see striations. It's the striated muscle. And lots of, inside, lots of nuclei. This is a multinucleated cell. And inside, there'll be a specialized organelle called a myofibril. Right? Lots and lots of these myofibrils filling up the entire length of the muscle cell. That's skeletal muscle. Okay. Now, we're going to dive into the muscle cell. Remember that a muscle cell is also called a muscle fiber. Okay, so keep that in mind as you're reading through this. Don't get those confused. Muscle fibers are very long. If you think about the sartorius muscle, right, pretty long muscle, goes around your thigh, that can be 18 inches long. So those cells within that muscle would themselves be 18 inches long and would be filled with these myofibrils, which also would be 18 inches long. These are very, very long, skinny cells. And what are those myofibrils made up of? Myofilaments, right? And the myofilaments, remember, come in two flavors, thick and thin. These are the actin and myosin proteins that you are at least somewhat familiar with. So if I look at this structure, this is the cell itself. I'm seeing lots of nuclei. Inside the cell, there are lots of myofibrils, right? And the myofibril itself will be made up of the myofilaments, actin and myosin. And that's what we're going to be talking about today, what's going on with this. Looking at this cartoon, I'm noticing that there's something else inside this cell that I've never seen before. And we'll be talking a lot about this. There is a specialized organelle in muscle. In addition to these myofibrils, there's also this structure called the sarcoplasmic reticulum. We'll talk a lot about it here in a second. But I want to introduce it right now because we can see it. It's this blue stuff, right? And the sarcoplasmic reticulum sounds a lot like the endoplasmic reticulum. And what the sarcoplasmic reticulum does for the muscle cell is it stores calcium. It is the calcium reservoir for the muscle cell. We talked about the importance of calcium in action potentials, right? Calcium is necessary for the exocytosis of the synaptic vesicles filled with the neurotransmitters. And I've said to you along the way that calcium is critical for muscle contraction, and we're going to be tired of talking about calcium here really, really soon. We're also going to see that sodium and potassium aren't going anywhere. We're going to be talking about the need for sodium and potassium levels to be accurate or correct for muscles to contract as well. But this blue web, what does reticulum mean? Reticulum, retinaculum, retina, reticular fibers. Network, right? So what are we seeing? A network, this blue is a network of plasma of membranes, right, within the sarco, within the muscle cell. And again, we're going to see that this stores calcium. And the muscle needs to have calcium available to it, so it has to store a lot of it inside the muscle cell itself. Then, so what we're looking at then, each one of these things sticking out is a myofibril. So that whole thing is a myofibril. And what that myofibril is composed of is primarily the thick and thin filaments, okay, actins and myosins. Now, in 105, I introduced very quickly that when you are looking under the microscope, you see striations in skeletal muscle. And what is it that causes those alternating light and dark bands? What causes the striations? What are we seeing? What's causing those striations seen in muscle? Cardiac and skeletal both have striations, right? Overlapping what? Absolutely.
Each other?
Overlapping thick and thin filaments, right? So we're going to see that those light and dark striping pattern striations are actually different overlapping densities, numbers of actin and myosin myofilaments, okay? So each of these little sections within the muscle where these actins and myosins are overlapping is referred to as a sarcomere. Sarco, muscle, mere. What does mere mean? Mere or mer? A polymer is a molecule made up of many parts. So mer or mere means part. So a sarcomere is simply the little functional part of a muscle cell. This is where all the action's happening. This is where all of the contractile force is coming from when a muscle contracts. Within a myofibril, there'll be many as 10,000 sarcomeres down along the length of the myofibril. Each sarcomere is very small. We're talking micrometer distances, right? So micrometer 1 to the 10 to the minus 6, right? So we're talking very, very microscopic structures here. I know that sometimes it's hard to imagine these very, very small things, but there's some really good pictures that will help us with this. And those overlapping zones are what we see as striations. Now, one sarcomere is defined as going from Z to Z. So the ends of each sarcomere are outlined by the Z lines. Some books will call this the Z disc, the Z line. It's a protein, okay? And it's going to be the outer limits, the outer edges of the sarcomere. And then there's an M line. Well, M in the middle, okay? So the M line is going to be right down the center of all the thick filaments. Picture's coming. The A band represents really just the length of the myosin molecule. Okay. The I band represents the area where you only have thin filaments, and the H band we'll see around the M line contains only the thick filaments. So let's take a look at a picture of this. We're looking at one myofibril. There would be thousands of these stuffed into a muscle cell, running along the entire muscle. And here's a Z line, and here's a Z line. So there's a sarcomere from here to here, and there's another Z line. Well, it's kind of cut off, but there'll be another sarcomere, another Z line right about here. So there's a second. Okay, so we've got two sarcomeres here, basically. And again, we see Z to Z, so Z to Z. Right down the middle is the M line, okay, M line. On either side of the M line, there's an H band. There's a better picture coming in a moment. The A band represents just the length of the myosin, okay? So the myosin is just the A band, and the I band is where you have the thin filaments, the actins. I wish the A band was actin. That would make more sense, but it's not. I didn't make this stuff up, right? So the way I remember this is the A band is the myosin. The I band, I is a skinny letter, right? That's the actin, and actin is a thin filament. So I have to kind of remember the shape of the letter. So the letter I is a thin letter, that's the thin filament, and the A is a broader, wider letter, so that is the thick filament. So what we're looking at then, when you look under the microscope and see the striations, you're seeing different arrangements of these overlapping A and M bands as the muscle is in different stages of contraction. I mentioned the sarcolemma already, right? That is really just the plasma membrane, the cell membrane of the muscle cell. And this is a brand new structure for you. So T-tubules, this is not something I discussed in 105. T-tubules we're going to see are continuous with the sarcolemma. I think of T-tubules as rabbit holes. Along the outside of the muscle cell, there are these little holes. Those are the T-tubules, and it's like a little tunnel that goes into the muscle cell. So the T-tubules, a tube-like structure. And we're going to see that it goes out to the sarcolemma and goes down into the sarcoplasm, into the cell membrane of the, or down into the cytoplasm of the muscle cell. And what we're going to see is that when the electrical signal comes from the nerve, right from the neuron, and that signal comes down and touches the muscle and tells the muscle to contract, that signal is going to travel along and then down the rabbit hole. It's going to travel down into the T-tubule. And we'll see all this in videos, and we'll get this story all put together here in a minute. The sarcoplasmic reticulum, what was that? That was that blue network right? And I told you it did what? It stored and held calcium. So that is also a very specialized structure. It's similar to the endoplasmic reticulum, right? And muscles do have an endoplasmic reticulum, but they also have this additional web of membranes called the sarcoplasmic reticulum. And when I show you a picture in a moment, we're going to see that the sarcoplasmic reticulum, along with the T-tubule that I had a moment ago, is going to form a triad. There's going to be three of these things side by side, and that is going, there's a triad, and we're going to see what this does. It's going to be a pair of the cisternae next to the T-tubule to form the triad. What's a cistern?
Cavern.
Cavern. When you hear about cisterns, anything come to mind? Cavern? It's not a bad way of thinking about it. We're going to see there's these two cavernous spaces, these two areas of sarcoplasmic reticulum right next to the rabbit hole, right next to the T-tubule. So that triad, and we'll see the significance of that as we go through this. So here's the cartoon of this. This is the sarcolemma, right? So that's the outside of the membrane of the cell. This is a myofibril. And wrapped around this myofibril is this network of sarcoplasmic reticulum. And the yellow is the T-tubule. Remember I told you that I always think of these T-tubules as little holes? Right, so here we are on the outside of the cell membrane, and there's these little holes. That's what you would see from the outside. But that little hole would open up into a tube. There's your T-tubule. Okay? And on either side of the T-tubule, I see two large parts of the cisternae. Okay, so that's creating your triad, right? You've got cisternae, T-tubule, and then cisternae. Those three parts are coming together. To me, it always looks like a little coin slot, right? I've got these two little sections with that little T-tubule down the middle, and that is forming the triad. I'm just introducing the anatomy of this, and then we'll figure out why we care about this. So that's the T-tubule. It's wrapped around, the sarcoplasmic reticulum and the T-tubules are wrapped around the myofibrils. So if that's a myofibril that it's wrapped around, what are all those little things in there? Those little dots in here would be what? The myofibril itself is composed of myofilaments, right? So those little dots would represent actins and myosins, right, within the T-tubule. So what does the sarcoplasmic reticulum do? It stores calcium. That's its major job. And it's going to pump calcium into the cytoplasm or the sarcoplasm as needed. Now, you're going to see that when that calcium goes into the sarcoplasm, it's going to interact with the thick and thin filaments and cause them to contract. Okay, so we're kind of starting to put this story together. The calcium will be necessary for the actins and myosins to attach and to pull and for the muscle to contract. Look at this. This is kind of a cool, just to get an idea. The concentration of calcium inside the sarcoplasmic reticulum versus that out in the cytoplasm, right, or the sarcoplasm, is a 40,000-fold difference. That's a pretty concentrated little silo of calcium right inside the sarcoplasmic reticulum. And when it opens up, there's a lot of calcium that floods out into the cytoplasm, and we'll see what happens. When a muscle begins to contract, remember we're talking about skeletal muscle here, it's voluntary. That means that a signal came from your brain, right? Came down from what part of your brain? The motor strip, right? We've been through this, right? Remember the motor strip, frontal lobe, right? Think about it. Coming down, and it's going to come down the spinal cord, and then that signal is going to go out one of the spinal nerves and tickle, well, I'm going to say tickle, right? Make contact with, what neurotransmitter would be released? Acetylcholine, right? Acetylcholine runs all muscles. And acetylcholine would be released and would go across the cleft, but here, on the other side of the cleft, there would not be another neuron. There would instead be a muscle cell. Where that neuron comes in close contact with the muscle, we're going to call that a neuromuscular junction, NMJ. Okay, and we're going to be talking a lot about the NMJ and a lot about how this whole thing works over the next couple of days. So as that signal, as that action potential comes down the axon and eventually causes acetylcholine to be released and eventually tickles or excites the muscle, we're going to see that calcium is going to be released from the sarcoplasmic reticulum, and that calcium will be critical for the contraction of the muscle. So guess what kind of gate is going to open on the sarcoplasmic reticulum to release the calcium?
Sodium gated.
Voltage or ligand gated? It's a calcium channel, right? When this thing opens, when the sarcoplasm opens up, it's going to release calcium. So it's a calcium gate, yes? And it's going to respond, what did I say? To a voltage, right? The action potential is going to come down to the muscle cell, and it's going to cause calcium to be released from the sarcoplasmic reticulum. So we would call that a voltage-gated, right? A voltage-gated calcium channel. Where else did you see a voltage-gated calcium channel? At the end of the axon, right? Remember, it was a voltage-gated calcium channel that opened up down at the synaptic knob as the action potential came down. It was a voltage-gated calcium channel that allowed calcium in and then bound to the synaptic vesicles and caused an exocytosis of the acetylcholine, right? So we have a voltage-gated calcium channel there, and now we see another voltage-gated calcium channel. This one is going to, when triggered, open up and release calcium from the sarcoplasmic reticulum, where it's very concentrated, out to the cytosol, where it is not as concentrated. Had a couple students this week ask me for, Mac, what's one more thing I can add? What's another way I can study? I feel like I'm not quite making all the connections. And I always fly right by these little review questions, don't I? If I were going to give you a study guide for this exam, I would probably just go back and pull all these review questions together. So don't blow these off. I know I blow them off. It's just there for you. But make sure you're looking through these and you can make sense of these. How many of you are actually looking at those, honestly? Okay, how many were just like, oh, he skipped it, I'm going to skip it too? Okay, thank you. Again, if I were going to give you a study guide, that's what I would give you, right? Can you define a transverse tubule? Can you tell me what's in a sarcomere? Can you tell me where the calcium would be greater in concentration? So make sure that you're asking yourself those questions and answering them. Maybe when you're reviewing the notes again, you're stopping and answering those questions right on the PowerPoint note. But don't ignore them. I think you'll find that many of the questions are directly related to those study objectives. Okay, two kinds of myofilaments. This is somewhat review, actin and myosin. And let's start off with the thin filament, actin. Now, it's not just actin, and I'm going to tell you a little bit more to this story than what I presented to you in 105. If I told you everything last semester, we wouldn't have to be here now. Okay, so I had to hold a little bit back. So the thin filament is, or has, a lot of actin in it. It's primarily actin. The thin filament is going to be attached to the Z-lines. What were the Z-lines? Each end of the sarcomere, I told you Z-line or Z-disc is a protein. The little thin filaments will be attached to the Z-disc or the Z-line through a protein called actinin. Okay? I wish it wasn't so close, right? So you have actin and actinin. Now, what we're going to see is that actin looks like a bunch of little beads on a string. Each of those individual little balls is called a G-actin, and it's called G for globular, right? Globular, a little glob, a little ball. So each of the individual little balls is a G-actin, but when I string them all together as a filament, we call it F-actin. So the G-actin are the individual little balls that you're going to see in a moment in the cartoon, and the F-actin refers to the filamentous or a long group of these G's linked together. So you'll see G-actin, individual molecules linked together to form the F-actin. And what holds the F-actin together is nebulin. Okay, so we've got more players, we've got more proteins in this thing than I have presented to you before. In addition, those are structural proteins. Okay, so actinin, actin, and nebulin are structural proteins. In addition, on the thin filament, there are two other proteins. They are considered regulatory proteins: tropomyosin and troponin. Okay, tropomyosin and troponin. And you'll see the picture in a moment, and you'll hear me talk about these enough. You'll be tired of them soon. But the tropomyosin, we're going to see winds around the thin filament, like a cord along the whole thing. And attached to the tropomyosin, we're going to see another protein called troponin. Troponin is going to look like a little snowman, a 3-part snowman in some of the cartoons. And there's 3 parts to this troponin. There is one part that binds to calcium, so that's going to be called troponin C. There's one part that binds to the troponin, or the tropomyosin, that's T. And there's going to be a part that is going to hold it all in place. So let me show you all this in a moment. I'm looking down here at the bottom picture. It's not very big, I know. So this is the thin filament. This whole thing is the thin filament. But there's actually quite a few proteins that make this thing up. So what do we see here? The little circles, the little red circles, each of those is a G-actin, a globular individual actin molecule, linked together by a protein that's not even shown here. There it is. A protein called nebulin. Nebulin is going to link, it's like the string going down the middle of the beads. And when all those little G-actins are linked together by nebulin, we now call it F-actin. Wrapped around this whole helix, the F-actin is like a helix. It's 2 strands wound together. Wrapped around it is this bluish-green cord. That bluish-green cord is tropomyosin. Okay? So tropomyosin is that cord-like structure you see wrapped around. And then troponin are these little blips attached along the way. Okay, we'll break that down. But troponin are these little proteins attached to the tropomyosin. Collectively, that is a thin filament. When you look at a sarcomere, let's go back up here, where would these guys be? Let me help you with this. This blue guy right here along the middle, that is the thick filament. So what band would we call that? From here to here, that'd be the A band, right? That's the thick filament. That's how long the thick filament is. The thin filament is this pink guy right here. There's one here, right? There's one here. See them? Those are thin filaments. They're attached to the Z line through that protein called actinin. Okay? So they're attached to the Z line through actinin. And if I could zoom in and look at that little thin filament, this is what I would see. Right? I would see all these proteins wrapped together to make the thin filament. So we're learning parts. We're just learning anatomy right now. Here's another picture of that. The thin filament is attached to the Z-line protein through this connector called actinin. So there's a nice picture, right? Nice picture of the whole. That's bigger for you. There are the players and pieces of the thin filament. So this is the thin filament all together. So it's really not fair to say that the thin filament is just actin, is it? It's really actin plus tropomyosin plus troponin plus nebulin. There's a lot of pieces that make up the thin filament. Versus the thick filament. It's thicker, it's bigger, that's why it gets that name. It is primarily made up of myosin, but we're also going to see that in the very center of the thick filament is the largest protein known in the human body. And that's why they called it titin. It is a huge protein. So they called it titin. Right? That's where it got its name. And we're going to see that it is the titin that connects the myosins to the Z-line. So what connected the thin filaments to the Z-line? Actinin. And what's going to connect the thick filaments to the Z-line? Titin. And titin is like a big coil. It's almost like trampoline springs. And in a lot of pictures, it'll be shown as like a coiled protein. And it's very, very strong, very, very big, very elastic. It recoils the sarcomere. So let's figure out what the myosin looks like up close. It too is a twisted, right? Each myosin molecule is actually two twisted myosins arranged such that the little golf club end of it is facing the thin filament. And we're going to see that it's the myosin that's going to attach to the actin. And this thing is going to pivot. So here it's at 90 degrees, but this head on the myosin molecule can also go up. So you're going to see that this thing can pivot about 90 degrees. And that pivoting of the myosin will be important as these molecules interact with one another and as they are pulling the filaments over each other. So let's take a look at this myosin. So this is the thick filament itself. It's a bunch of myosin molecules. Okay. And we see that going right down the center of this myosin, right down the center, we can see just a little bit of it sticking out. This is that spring-like titin protein. But imagine it going straight through the middle of the myosin all the way across. So there'll be more titin over here. Okay? So it's like the very center of the whole myosin. And what would be right here? Right. So the titin is going to attach the myosin to the Z. And there'll be another Z over here. Okay? Again, how long the myosin molecule is defines the A band. Okay, so you're going to see that a lot. Now, what do these thick and thin filaments do? Right, we're talking about the microstructure of a skeletal muscle. We'll see also these thick and thin filaments in cardiac muscle. Okay, because remember, cardiac muscle is also striated, so we're going to see the same sort of microarchitecture when we get to cardiac muscle. What do these actins and myosins do? We've heard this word before. They slide over each other like little molecules playing tug of war. And when they pull on each other, what happens to the sarcomere? The sarcomere will get shorter, right? So each of those little units will shorten. All of the sarcomeres along an entire myofibril will shorten simultaneously. And as a result, the entire muscle will contract. That whole muscle cell will contract. So this is sort of an all or none type thing. All of the sarcomeres in all of the myofibrils of one muscle cell will all simultaneously begin to contract. Some sarcomeres don't get to choose to contract and not others. Every sarcomere within every myofibril of the entire muscle cell will, when activated, contract simultaneously. And all of those millions of myosins and actins now sliding over each other are going to give you the power and the force that are generated by muscles. When someone works out and their muscle enlarges, what you're actually doing is increasing the numbers of actins and myosins and more myofibrils. And what that's going to do now is give more power, right? More overlapping actins and myosins to work together. When you take the sarcomere and you imagine it is being activated and it is shortening, what happens to the H, I, A, and M, and Z stuff. So when you look at this again, when the thin and thick filaments slide over each other, you will notice that the H band and the I bands are going to get smaller. They're going to become less obvious or almost disappear altogether. The Z lines will definitely get closer together, right? The two ends of the sarcomere will close in on each other, and the entire sarcomere will get shorter. Will the A band change? What is the A band? The A band is as long as the myosin molecule is. When the actins and myosin slide over each other, are those molecules themselves shortening? Have we seen any evidence that actin and myosin are shortening? No, they're just sliding over each other. So the A band won't change because myosins are still myosin. They're not changing their length. So as we look at this, we'll see that the A band will stay constant. The overall myofibril will get shorter. The entire muscle will get shorter. But the filaments, the actins, and the myosins themselves do not change length. They simply slide over each other. So here is a sarcomere at rest. Again, Z to Z. That's a sarcomere. Thousands of them, 10,000 of them, lined up end to end along a myofibril. Within that, there's the A band. The A band represents what again? The width or the length of the myosin. So if you compare that picture with this one, where we have now a contraction, that A band will be the same. Right? It has not changed. What has changed from left to right? What has gotten smaller? Look at the labeling things on the right and look at them on the left, and what has changed? The sarcomere has gotten shorter, right? If I were to measure this, the sarcomere is shorter. How about the H band? What was the H band? The H band was this kind of lighter area in here. And what happens as the muscle contracts? Those actins are sliding over the myosins. The H band really only represents that area where there's only myosin. Okay, so we see that gets smaller because the actins are cringing in the center. The I band wasn't even shown here, but the I band will also be getting shorter. Well, actually, I can put it in here. There it is. There's your I band, right? It's marked over here. The I band is where just the actins are, right? So what's happened over here? It also has gotten a lot shorter, hasn't it? So the H and the I got smaller. The sarcomere got shorter. The Z to Z distance got shorter. But the A stayed the same. Okay, so keep track of those little members as we move through this conversation. So, thinking about this, this is one myofibril. It has been activated, and here we've got how many sarcomeres do we have here? 1, 2, 3, 4, 5, at least 6 or more sarcomeres are shown, and they have all simultaneously shortened. So when this happens, this is an all or none phenomenon. All sarcomeres, all myofibrils within a muscle will all shorten when they're activated by a neuron. So how does that work? Okay. Oh my, we're back to membrane potentials. You thought it was gone. Because we've already been through this story, I don't feel obligated to tell you it in as clearly or as laid out as before. Sequentially, this is chapter 9. Muscle comes before nervous system. So Martini and company have spent a little bit of time here. If you went through this book sequentially, you would not have yet talked about action potentials, and you would not have yet talked about sodium and potassium and excitable membranes, and you wouldn't have talked about depolarizing and hyperpolarizing and all that story. We know that story well, right? We're going to dust it off. We know that story well. That story is no different here than it was in neurons. Neurons are an excitable cell. Muscle's an excitable cell. And by excitable, it means that there's a difference in the membrane potential, and it can respond to electrical signals. So we know that muscle cells are polarized, okay? Which means what? On the inside, it's more negative. The outside, it's more positive. That hasn't changed, right? Just like we learned back in neuron world, we learned about negative on the inside and positive on the outside. And guess what? The sodium and potassium are still in the same place. Nothing has changed. This is not a new story. It's just a reminder of a story we already know. The resting membrane potential of neurons, remember, was negative 70? It's so nice. It's also negative 70 for skeletal muscle. So nothing has changed here. Okay. As a quick reminder, if I could take a little electrode and stick it inside a muscle cell, I would see that at rest, the cell has a negative 70, meaning there's more negative charges on the inside of the cell than there are on the outside of the cell. The extracellular fluid is more positive. It's more negative on the inside. Remember, the major ion out here is sodium. The major ion inside was potassium. That's not going to change. That story is all the same. If you got it last time, you're feeling comfortable. If you didn't get it last time, here's your chance. So what is it that's going to contribute to this polarity? Same players. That's why I'm not going to spend a lot of time here. The positive charges, sodium and potassium. Sodium is mostly in the extracellular fluid. Potassium mostly on the intracellular fluid. The major negative contributors are chloride ion, mostly where? Wherever sodium is, chloride is found. So sodium is on the outside. That's where chloride is going to be found as well. And within the membrane, within the cell, there are a lot of proteins that can't move in and out. And so there are a lot of very large negatively charged proteins inside your cell. What else is negatively charged inside your cell that creates some of that negative value inside the cell? It's not mentioned here, but what else is negative? What other molecule is negative that is found inside the cell?
Proteins.
Proteins are mentioned. Most proteins are negative. Right? Many proteins are negatively charged, but right? We know DNA and RNA. We know a little bit about them. Remember, DNA and RNA, they're negative. What's on their backbone? Phosphates and sugar. Phosphate, sugar. And that phosphate is negatively charged. So DNA and RNA are also negatively charged. That's just contributing to the overall negativeness, if that's the right word, of the inside of the cell. Oh, this isn't any different, right? We're going to have movement of sodium and potassium. If there were leaky channels, in other words, it just kind of happens because of concentration, we would expect sodium to try to leak in, right? Flowing in. And we would expect potassium to try to leak out. And that happens. But who controls the Who controls it? Yes, there's leakiness of sodium and potassium, but what pump is going to make sure that everything stays right? Sodium-potassium pump, right? And that sodium-potassium pump does what? Sends 3 sodiums out for every 2 potassiums that it allows in. So that's also helping to maintain that negative charge on the inside of the cell. Check. Been here, dusted off. Remember this. So here's a picture of that. There are sodium leak channels. Sodium would try to come in. There are potassium leak channels. Potassium would try to go out. What's not shown here is another protein, right, called the sodium-potassium pump. And what does it do? 3 sodiums back out for every 2 potassiums that come back in, right? So that's going to, again, keep that negative charge on the inside. So there are voltage-gated channels that are going to contribute to the membrane potential. In the previous picture, these are leak channels. That means that they're not responding to voltage changes, right? These are not opening because of a voltage change. They're simply leaky channels. Sodium gets in, potassium gets out. It just happens. But remember that in addition to that, there are voltage-gated channels. And these are the ones we talked about with action potentials, right? The one that opened the sodium for the depolarizing, that was a voltage-gated sodium channel. And the one that opened in response to the potassium was also a voltage-gated channel. So this is going back to the world of action potentials. Okay? And action potential is always the same. All right, nothing has changed here. It's the same story. So there are voltage-gated sodium channels that open. The graph goes up to positive 30. Remember this, right? Goes up to positive 30. And then we'll come back down. That's repolarizing. It will always overshoot a little bit and hyperpolarize. And then it will come back and become stable again as the, it'll come back and get stable again as the resting membrane potential. There is a little bit of time necessary to restore the sodium and potassium, and that's called the refractory period. We saw that, right? We know that. We know that action potentials have a refractory period. There's a little bit of time where the sodium and potassiums have to get back in their proper place before another action potential can be fired. So a quick reminder, I'm not going to ask you to write this out again like you did last time. This is something that you should be able to kind of draw out on a piece of paper. It's just a reminder, because skeletal muscle is going to be activated by an action potential. So I just want you to thinking about what that means. And then we're going to see that the muscle is also going to pick up the signal and cause the muscle to contract. So we need to go back and remember a little bit about action potentials. So what do action potentials do? We know this as well. They travel down the axon. They go very, very quickly. They go faster if it's myelinated. And I'll tell you that your skeletal muscles are responding to myelinated axons. So this is all very, very fast. You think it, the signal is going to zip down your spinal cord and out your neurons. Those are all myelinated, very, very quick responses. And this is all in very, very short time. Okay, we know this. We can use an oscilloscope in lab tonight, Thursday night, Wednesday night. We're going to be doing a little PhysioX experiment. It's an online activity to look at action potentials in lab. You guys should do really well in this because you've already had action potentials. And so when we go over that in lab this week, it should be very much a review. The instrument that allows you to see the blip on the screen, that allows you to see the action potential, is called an oscilloscope. Okay, so don't be thrown off by that word. So the oscilloscope is the instrument that allows you to see the little blip of the characteristic line of the action potential. When that action potential comes down the axon terminus, causes the voltage change, the voltage-gated calcium channels to open, causes the exocytosis of acetylcholine. Acetylcholine wafts across the synaptic cleft, and then, in this case, is going to interact with a muscle cell. That is going to be called the NMJ, the neuromuscular junction. Before, we talked about one neuron speaking to another neuron. Here we're talking about a neuron talking to a muscle cell. So this is the NMJ in this chapter. And this is important. There's only one NMJ per muscle fiber. There's only one NMJ per muscle cell. It's kind of interesting. What am I telling you? That sartorius muscle that wraps around your thigh, those cells are 18 inches long, right? But only in one place along the length of that 18-inch muscle will there be one neuron touching it. Okay, so only one place along that cell. So every cell will only be attached to one neuron. Okay. Now, in lab 2 or 3 weeks ago, in a pre-lab for muscle lab, you defined, do you remember this, a motor unit? And that was kind of like, what's he talking about, right? For you guys especially, because we hadn't talked about it. Do you remember what a motor unit was? It's one neuron and all the muscle fibers that it communicates with. Okay? So a motor unit, okay, is one neuron and all of the cells that it attaches to. So this is what this is saying. One motor neuron may branch to control multiple muscle cells. Another way of saying a motor unit. Okay, so motor unit, one neuron, and all of the muscle cells that it innervates or touches. These are great little videos. There's some really nice ones. If you wanted to watch these, you can do so, by going just to the PowerPoint and opening up in presentation mode. These AP Flicks videos are also available to you in the study area of mastering. Go to mastering, go to the study area, and then on the lower left, you'll see Lab Flicks, okay, or AMP Flicks, these little movies. So I think you'll really benefit from watching these. I'm describing everything that it shows here, but this is a great tool for you to really get this down. You know what, though? I'm going to go ahead and show it, because I think it'll be helpful. I don't know if we're going to hear it. There's always issues with that, but we'll show it.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps.
We know these.
Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels.
Usually sodium, right?
Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma.
Pretty familiar, right?
Transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
Okay, so we know those steps. The only thing I would say to that is when in the neuron, when the acetylcholine went across the cleft and into the dendrite or the soma of the next cell, what would be formed? An action potential or a local potential?
Local potential.
A local potential, either as an EPSP or an IPSP. Remember this conversation? Either an excitatory or an inhibitory signal. That's the only difference here, right? So once the neuron comes down and touches the muscle, they're going to describe it as being another action potential because we're not in a neuron anymore, right? We don't have to wait to get to the axon hillock to cause another action potential. So when threshold is reached on the muscle, quote, an action potential is generated. So it's a little bit different than what we discussed back in neuro world, but not significantly so to throw you. Okay. Now, there's another video coming up, and we'll watch that one as well. So what's going to happen is this is just a cartoon step-by-step of this. But here are the vesicles containing acetylcholine. It's going to go across the cleft, and it's going to cause the movement of sodium into the cell. Now, the next video is going to show this better. That action potential, she told you just a moment ago, is going to travel along the sarcolemma. What is that? It's going to travel along the cell membrane of the muscle cell. And what she didn't say is where that signal is going to go. Anybody have a guess? I've already mentioned them. What are the rabbit holes, right? Those little rabbit holes that are along the membrane of the muscle cell. That electrical signal is going to go zip down that rabbit hole. What's on either side of that rabbit hole? That rabbit hole was a T-tubule, which formed a triad. And on either side of that triad, what was there? Two cisternae. Two big parts of the sarcoplasmic reticulum. And what was on the wall of the sarcoplasmic reticulum? Voltage-gated calcium channels. So we're starting to put it together. So the action potential comes across and zips down, right? That voltage change zips down the rabbit hole, down the T-tubule. That voltage change is now going to do what? Open up the sarcoplasmic reticulum and release calcium where? Into the sarcoplasm or into the cytoplasm of the muscle cell. Are we following that step? Now what we're going to see in a moment is what does that calcium do? We're going to see in a moment how that calcium is going to bind to actinomycins, we'll see, and cause contraction of the muscle. And once you can pull all these little pieces together, you have, boy, a big part of the next test. Right? A big part of the next test is going to be understanding this little story. And I think, hopefully, you're going to be convinced that you already know a lot of it. So at the neuromuscular junction, another thing that's a little bit different, at the neuromuscular junction, you've got the synaptic knob. We can picture that. And what it touches on the muscle or what it synapses with is called the motor end plate. That's a new word for us. So the synapse is the axon terminus coming in close proximity to the motor end plate of the fiber. And that area has all the acetylcholine and acetylcholine esterase, that can break down the acetylcholine. That action potential that is all that sodium that rushes in is going to cause another depolarization of the muscle cell. That action potential is going to run along the outside of the muscle cell. Imagine it's sort of similar to how an action potential runs along an axon. Okay? So it's going to run along the outside of the sarcolemma, around the outside of the muscle cell, and then that electrical signal is going to dive down into those T-tubules. As long as acetylcholine is being released, then sodium will be flooding into the cell. And as long as there's sodium flooding into the muscle cell, there'll be action potentials generated along the muscle. So as long as there's acetylcholine being released, there would be calcium channels opening in the sarcoplasmic reticulum. The moment we take away the acetylcholine, in other words, stop the action potential coming down the axon, then quickly acetylcholine is wiped away, and those calcium channels will close up on the sarcoplasmic reticulum, and the muscle would then once again relax. So we'll put all this together. I've already mentioned that. This story is called excitation coupling or excitation contraction coupling. In other words, we've got to pull together two important ideas. We know about the action potential. We know about the excitation of the membrane. And then how are we going to get from excitation of a membrane to the muscle contracting? This they've simply put together in an awkward phrase called excitation contraction coupling, bringing these two ideas together. And we're going to see that calcium is a big part of what connects or couples these two activities. So let me phrase it. Here it is, right? The action potential comes down to the motor end plate, moves across the sarcolemma. That action potential will move down the rabbit hole, down the T-tubule, right in between those two terminal cisternae, those two little pockets of calcium. And remember that, what do we have there? Voltage-gated calcium channels. This is truly the coupling. How do we go from an action potential to causing contraction? It is all about calcium. It's all about the sarcoplasmic reticulum. So really what you're doing is by allowing calcium to be released from the sarcoplasmic reticulum, you're going to cause the muscle to contract. So that is the excitation, contraction, coupling of this whole story. It's an awkward phrase, but when you put the words together and realize what it's saying, it's awkward, but it makes sense. Let's take a look at this one as well.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with a calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
Not too bad, right? Putting it together. It really is a story. We've got to be able to tell the story. I think we're getting closer. So this cartoon is just showing the action potential is traveling down along the sarcolemma and jumping down in that rabbit hole, right? Down that T-tubule. And on either side, what do we have? Cisternae filled with calcium. The voltage of that signal is going to open up those calcium channels, and that calcium is going to flood out into the cytoplasm or the sarcoplasm of the cell. So, what does that darn calcium do? Okay, so here's where we bring in the actins and myosins again, and we figure out what they're going to do with this calcium. The sarcomere is at rest. Can you picture it? When you picture a sarcomere, what do you picture? You're going to be dreaming about this stuff really soon. Okay. So, sarcomere, right, from Z to shining Z, right? From Z to Z. And what do we have in here? Some myosins and some actins, right? And so at rest, they're kind of overlapping a little bit like that. I'm not drawing in the nebulin, I'm not drawing in the actinin, but you can see that that's a sarcomere at rest. Now, what I can't draw in is that at this point, the myosin is, quote, energized. We'll see what that means in a moment. And is cocked, is pointing away from the M line. And we're going to see that there's some ATP involved here. It makes sense, right, that moving muscle is a very energy-necessary step. So we haven't talked about ATP, but clearly there's got to be some very important ATP requiring steps in this. And we're going to see that the myosin head itself, the protein head of the myosin, that big golf club, is itself an ATPase. What does that mean? ACE, an enzyme that breaks down ATP. So ATPase, an enzyme that breaks down ATP. So this ATPase is going to break ATP down into its parts. ATP, when it gets broken down, becomes ADP plus phosphate. What does ATP stand for? Adenosine triphosphate. There are 3 phosphates. When we break it, now ADP is adenosine diphosphate. So you've taken one phosphate off, released a phosphate, you, quote, inactivated the ATP. It's no longer able to do what it does. This is too small for you to see, I know. I didn't mean to put a small one in there for you. But what we're going to see, I can make this bigger on my screen, but this doesn't make it any bigger for you. The calcium is going to come in. Do you agree right now? Hold on, I'm going to look at something. Yeah, I thought I had a better picture of this. Let me try to do this for you, and then we'll watch the little video. So the calcium will be released from the sarcoplasmic reticulum. It's going to come out into the cytoplasm of the muscle cell. What is the cytoplasm or the sarcoplasm filled with? The sarcoplasm is filled with those long myofibrils made up of sarcomeres. And on the actin, what did we have? What was that protein that wrapped around the F-actin? Right? So tropomyosin was that cord-like protein that wrapped around the helical molecule of actin. And what was on tropomyosin was that other molecule called troponin. So I'm going to draw it like a little snowman, okay, on the tropomyosin. Well, guess what? That calcium is going to bind to troponin. Okay? That's what calcium is going to do. It's going to bind to this molecule called troponin. Now, when it does that, it's going to cause this whole tropomyosin molecule to shift. And when that tropomyosin rope-like molecule shifts, it's going to allow the actins and the myosins to make contact with each other. And when they make contact with each other, they're going to start pulling and doing that sliding filament dance that they do. Okay? I apologize, I don't have a better figure of that here. Martini does break this down step by step, and I think I grabbed the wrong image for you for that particular figure. But we'll see this in the upcoming movie. So this begins the contraction phase. We've got calcium. The calcium was released by the sarcoplasmic reticulum. It's going to arrive at the sarcomere along thousands of sarcomeres. The calcium is going to bind to troponin, as I just said. And what that's going to do is cause the tropomyosin molecule to shift its position. The shifting of that tropomyosin is now going to expose myosin binding sites for actin. When tropomyosin is there, actin and myosin can't see each other. Okay, so when tropomyosin is in its normal position, actin and myosin are unable to connect one to another. When calcium's around, it binds to troponin, causes the whole thing to shift, and now the actins and myosins can attach. And as they attach, we're going to see that it's going to start moving. That little cocked head of the myosin is going to start moving, and it's going to start pulling those filaments over each other step by step. So that myosin head's going to pivot. And as it pivots, it's going to, we call that a power stroke. And we'll see this in the video in a moment. And the ATP is going to be broken down, and it's going to detach, and then a new ATP is going to come in and cause it to release, and it's just going to keep going over and over and over again. As long as there is calcium, the actins and myosins will keep grabbing each other and pulling. Now back up. I said as long as there's calcium. What's going to make sure that there's calcium there? Calcium is there because there was a constant flood of sodium causing action potentials. Sodium was there because there was a constant flood of acetylcholine. Acetylcholine was there because there was a constant signal coming down from the action potential, right? So the neuron continues to send a signal, and this calcium continues to be available for the actins and myosins to do their little trick. And then we're going to see it goes back and forth. It recocks, resets, recocks, resets. So let's watch this movie. It'll do a far better job than my cartoon.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate.
That causes it to
The
stand
energy
up.
liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released, and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position.
Nice video.
As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
Nice little video. The only thing I don't like about it, well, what's difficult to show, is that it's not just one myosin talking with one actin, right? And in that one little image, it showed you, more of these myosins and actins. So you have to imagine in your head you've got thousands, millions of these actins and myosins all grabbing and pulling. Power stroke, power stroke, power stroke. The other thing you notice is that what did ATP do? ATP, when ATP came in, what did ATP do? Cause the actins and myosins to separate. So when we talk about rigor mortis next time, right, at death, what happens to the muscles? They stiffen. The actins and myosins get stuck in a contracted phase. What is it that's going to release actin and myosin? I just told you that it's ATP that normally will disconnect actin from myosin. Clearly, after 36 hours of death, your body does not suddenly find ATP in order for the actins and myosins to disconnect and to relax again. But instead, after 36 hours or so, the muscles are already beginning to break down. And the lysosomes within the muscles, they have lysosomes too, will start to release their enzymes and actually start breaking down actin and myosin molecule proteins and cause the release of those molecules one from another. So you'll see the ATP story pick up here quite a bit. But we basically right now have all of the pieces for this. This whole thing's called a cycle, right? A contraction cycle. And as long as there's plenty of calcium around, and as long as there's plenty of ATP available, and as long as you're sending your little action potential down your spinal cord to your motor neurons, you will continue to have muscle contraction. Once you stop sending that action signal, right? Once you stop sending that signal, once the action potential stops coming down, once you stop releasing acetylcholine at the motor end plate, once you stop causing that action potential, then you'll have no longer a voltage change and those calcium channels along the SR will close. Very, very quickly that calcium will be sequestered, taken away, sort of like how acetylcholine esterase got rid of the acetylcholine. There is a protein in the sarcoplasmic reticulum called calsequestrin. Okay, it sequesters the calcium, and it kind of cleans it up, just like acetylcholine cleans up the acetylcholine, acetylcholine esterase cleans up the acetylcholine. So I think the video does a nicer job, but the Martini does a nice job, and I forget which page it is, but that whole 2-page spread that goes through this step by step by step by step. I don't know if I will. I don't know. I haven't decided yet. On the next exam, there'll be a short answer question, but I can guarantee you the short answer question from this chapter would be, what are the steps involved with muscle contraction, right? And there's a lot of players here. And so you need to be able to sketch that out. Now, I may not ask it for points, but I guarantee you if you can sketch it out, you can answer questions about it. So likely, regardless if I ask the question on the exam for 5 points or not, you'll want to be able to turn over this next test and either know, like the back of your hand, these steps, or sketch it out quickly and refer back to it during the exam, and I'll be asking questions about the steps of this. So watch the videos, think about the steps, break it down in your head, and it'll really, really help you as you're preparing for this. I'm going to stop there. It's a good place to stop. That's a very important set of steps. Anything that you can articulate right now to clarify this whole excitation, contraction, coupling, contraction of the muscle, actin and myosin, thin and thick filament, anything in that conversation to clarify. We'll continue to talk about muscle on Thursday. I hope to finish up this chapter by Thursday. I'll put a quiz, like I said, on mastering about this over the break. And then when we get back from break, we'll be talking about cardiac muscle and the heart and EKG. When we get back from break, we'll also be doing EKGs in lab. So it'll be a nice time to come together on that material.